Yeah, and we're going to start off with the SCRI projects update. That's a specialty crop research initiative. People aren't familiar with that acronym. So there'll be 4 presenters going. There is a change in the, there was a cancellation, unfortunately. So it'll be Julie Urban going first, then Charles Bartlett, and then we'll move Tracy Lesky. Dorothea Toll was not able to make it. And so we have a last minute addition, like thankfully Kelly Hoover from Penn State agreed to do a presentation. So it might not be in line specifically with the SCRI research, but Kelly will provide information on the work she's been doing too. All right, thanks. So Julie, if you want to go ahead.
Great. Can you see my slides and hear me okay?
Yeah, yes to both.
Okay, great. Thanks so much. Thank you all for the opportunity to be here. It's like really exciting to see all the cool updates that are going on in terms of research extension and regulatory activity. And so as Greg indicated, we have a large multi-institutional specialty crops research initiative grant that's funded by USDA NIFA. And so that is what supports the stopslf.org website, where all of these talks are going to be recorded. And so please check that out. What I want to share with you today is, are some of the projects that my lab is working on, but also, so I want to talk a little bit about some preliminary data we have concerning looking at SLF survivorship and damage on different specialty crops. And then also stuff that really we've been fundamentally involved in looking at adult reproductive development and in a way to try to explain late season movement. But also I want to share with you other patterns that we're seeing with late season movement, now that we've been into this for 2 to 3 years and really trying to make sense of it all. And so of course it's, in this large collaboration, it's not just my lab that's trying to make sense of it all. We're all working very collaboratively, even with other folks who are not part of the SCRI, to try to make sense of these things. And in a sense, you hear these different stories, or you hear the results of any one study, and it might sound frustrating because it seems like perhaps these are, inconsistent sets of results. How do you come up with conclusions? Well, there aren't any easy answers with lanternfly, but I feel like we're really starting to weave things together a bit. And I'm super excited about where we're going, especially starting this next year. So let me begin. All right. With this issue of survivorship and damage assessment on different specialty crops. You all heard, if you were here yesterday, you heard Danelle Brown from the Grape Research Association, concerned about California, they grow so many different crops. So some of this work is funded by the California Department of Agriculture. And so basically what these are is just, trying to screen a variety of different specialty crops to see what lanternfly can survive upon. And then also, does that do any damage? And again, I'll come back to this at the end of my talk. This is kind of artificial. We're kind of force feeding lanternfly on different things, but at least it lets us do a preliminary screening of what crops might be more versus less at risk. And so Heather Leach started this work not last field season, but the season before, and then she's moved on to bigger things. And so my postdoc, Holly Shugart, and technician Elaine Gomez and I did another round of these studies. And so basically what we focus on overall, we're looking at survivorship of 4th instars or adults. Heather looked more at 4ths, we looked more at adults last year, feeding on a different variety of crops. So we counted these insects every 2 to 4 days and replaced dead insects to maintain that constant pressure on plants. And you can see we looked at peach, raspberry, cucumber, hops, grapevine, tree of heaven, and we'll look at more this year. And so this is a surprising result that really got our attention. And this was work that Heather did in 2020. And so she placed a fixed number of, she put lanternfly in varying numbers. We don't have many replicates for these studies. These are all very preliminary before we blow out into much bigger experiments. But basically, this shows you the kind of setup. This is out at our Penn State Berks campus, where we've been able to take over part of the turf research plot with all sorts of different plantings for lanternfly work. And what was surprising is that when lanternfly were caged on these cucumbers, you see that, higher numbers of spotted lanternfly decreased cucumber yield. So this is a really striking study. We're growing cucumbers in my lab right now. We're going to replicate this with higher numbers in the lab. One thing we did find this last year, though, Heather found really no detrimental effects of fourth instars feeding on peach trees. But when we put adults caged on peach trees, we got a little bit more of an alarming result this year. No fruit was presented. Shoot length wasn't influenced, but shoots exposed to lanternfly were yellow, you can see, and were defoliated, and they dropped their leaves. And so again, somewhat artificial, but potential impacts here with peach. And then the one that kind of surprised me a little bit, and was that John Rost and Heather planted hops out there a couple of years ago. And Heather found really no impacts to hops. And when we started this on hops, there were actually ambient lanternfly feeding on them anyway. And not in high numbers, but that kind of got my attention. It's like the hops really took off this last year. And so, control hops, after we're kind of force feeding lanternfly, we're caging them on. But basically, compared to controls with no lanternfly, they had no dead leaves or vine necrosis. If you put high densities of lanternfly on these hops, that's really, really going to mess them up. And so we're going to be following up with that. And again, I'll come back to more about this at the end of my talk. But basically, overall, we're looking at, are there we're not just looking at these studies alone, we're listening to growers, are there any field reports of damage or, populations of lanternfly coming on particular crops? And certainly we see it with grapevine and tree of heaven, but the cucumber, it is seen on cucumber. And yes, they seem to be able to live on it to survive, and it does reduce yield. So we're kind of ranking the relative risk here. And so you can see that, when fourths were tested, you had some different results that, compared to what we found in 2020 with adults. And also, are these viable hosts? It'd be great if lanternfly couldn't even feed on them, but a lot of these are really potentially viable hosts for lanternfly. And so again, we're continuing that work this year. And so I want to move on here to other things about reproductive development. If you got anything, if this is your first time in lanternfly discussions, or if you were in Spotted Lanternfly 101, the one thing we know for sure about lanternfly is that it moves, and it certainly moves seasonally across its life cycle. We saw it, in 2017 coming onto apple. Fortunately, it was transient. We don't see any damage. That's not really a problem. But in this late season, mid-September to beginning of October, that's when we see egg laying. Oftentimes, that's when you might see lanternfly moving into new areas. Certainly, that was the story that you heard yesterday in terms of like what Anne showed and what Doug Pfeiffer showed. And here these are, lanternfly eggs that were laid then as lanternfly moved on to red maple, and in a vineyard we studied. And so if you're here for Lanternfly 101, I'm going to talk a little bit more about the study that was done in Wyomissing, because this is where my dissections and work on female reproductive development focuses based on these specimens. Here we have Dennis Calvin and John Rost identified 20 similar age and size red maple trees. They were all kind of planted at the same time when this suburban development outside of Wyomissing was built. And so what they did is they put traps and then collected lanternfly every week throughout 2019 and 2020. So what I'm showing you here are my data for 2020 or for 2019, but basically what they found, and this is interesting in terms of this movement, by putting sticky bands, and so these are things that trap out lanternfly, by putting sticky bands on these trees and changing them out every week, you're seeing, okay, are more lanternfly coming in? No, it looks like they're not. It looks like these traps are kind of trapping them out and reducing population numbers, which is really exciting. But then, oh, look, adults move in, you're looking at, 20 trees, an average of over 600 per tree. And then when you say, okay, what about egg masses? Remember the 615, we have to move the scale because boy, if you count egg masses and estimate based on, 37 eggs per egg mass, you see, boom, you got a big bump, a big increase in that in that population size. Where are they coming from? Well, this is what we're trying to figure out. How do we explain this late season movement? Because if we can explain it, we can predict better the timing of when people should look for these and the management. So they're coming in, we're thinking that, okay, well, maybe they've depleted their Ailanthus hosts. And so, why are they coming in? And so again, we're kind of presuming that they move in because they need to feed heavily to complete reproductive development. This is when we saw them coming into vineyards. These are Heather's data based on, from 2018 through 2020. And so are they increasing their mass? Yes. When I've gone, and collected them at vineyards, taken their live weight out in the field, yeah, they're packing it on. Okay, so they're really they are feeding heavily, they are getting larger. That makes sense. And so you say, okay, if they're really feeding to complete their reproductive development, what influences the timing of insect development? Well, these are exothermic animals, temperature. So therefore, we've been working to develop a degree day model of lanternfly reproductive development. And so for this, what I'm showing you are 20 females per week that were collected, 10 from red maple, 10 from the Ailanthus in that neighboring area. And I looked at whether or not they had mated, basically I assessed how mature the eggs or the ovaries of the eggs were. And then as a proxy for that, we know these pretty little lanternfly yellow bellies, as they fatten up, get more visible, they expand more. And so we're taking those measurements to, those are going to be correlated with, and we're cranking these numbers, but we'll look at, the extent to which they're correlated with mass as well as with the internal development. And so for this model, for the results I'm showing you, we used a base threshold of 10.4 degrees Celsius. That came from the base threshold coming out of my student Erica Smyer's work on egg hatch that integrated data she collected with that from 2 studies done in South Korea. But if you look at Melody Kina's work coming out of her lab, where she's reared them, at different temperatures and looked at development of the different instars, if you average those base temperatures across all the different instars, it comes darn close to 10.4. And so this, if you took Lanternfly 101, you showed how do you tell if a female has mated? This is what the spermatophore looks like. So that's what the male gives to the female when they mate. And so if we look at this, just looking, again, these are the weekly lanternfly that I dissected from that Wyomissing study. And you can see those little yellow bellies get bigger over time. When did they start mating? Well, I found the first spermatophores here it was with that base 10 threshold, just over almost 1900 growing degree days. But although I saw the first egg spermatophore there, of those that I dissected, 40% were already mated. And you can see they're still moving in, right? They collected out everything every week. So the next week, by the next week at 1940 degree days, 85% of the sample had mated. And that's where we saw the first mature eggs. And so again, happened a lot faster. 35% of the sample had mature eggs. And then by that following week, 75% of the sample had mature eggs. 2020 was a very, very different year in terms of degree days. I'm working on that next. We think there might be some other cue going on, that it's not just completely driven by temperature, but that's something that Dennis Calvin's working on, and my stuff will follow up. But I do want to show you a couple other things in terms of trying to weave together these really weird patterns that we're seeing. What's going on here? Are they laying additional egg masses? Well, that's what I'm looking for and recording as I dissect. But also there looks to be like a good amount of variation. Okay, and so here I just put together a table from 2 studies that Hu Ping, you'll hear from later, did in terms of estimating, making observations of when the different life stages occurred in the field, and using a base 10C threshold, looked at what were the accumulated degree days across these different life cycle days and when they laid their eggs. And then Doug Pfeiffer presented some really awesome work that he and his student Andy Deshane did, where they didn't report the range of accumulated degree days, but they reported in that paper the calendar day. And so you see a really similar pattern in, in Virginia and in what Huping reported in Pennsylvania. At any given year, there's a huge degree of variation in the timing of development, where you have like 2 and 3 different life stages occurring at most times of the year. So if you look here in the 2016-2017 data, you have thirds still hanging around when you have adults. Okay, so there's a lot of variation there in terms of that development. And so then you say, okay, but why do we have these definite peaks? If everybody's kind of developing at a different time, why do we see, or do we see certain times when they're coming into vineyards? Okay, we saw this in Heather's data, 2018, 2019, that peak was in September. Well, at one of those same vineyards last year, where, Greg showed you that wall of exclusion netting, that peak didn't happen in last year in September, it happened in October. And we see these other weird patterns. This is a study that was published that Kelly's postdoc Charlie Mason led. And these are different sites where we had red maple and they were trees were treated. And we observed the number of lanternfly coming in, typically weekly. And so with this one particular site, you see that peak was at, in September. But these other sites, it was all like that peak was seen in October. And so you can say, well, all right, maybe the peak was a little bit earlier in 2018, and these down at the bottom were all 2019. But I want to point out this one site, this is Topton, and so here you see, we're just talking like an average of 30 lanternfly coming into these trees. Well, this is a site that Brian Walsh, who you'll hear from tomorrow, he has some awesome data. He's been testing different ornamental treatments for us. He's continued to monitor. And so he shared this with me so I could share it with you. That if you look, this is that exact same Topton site, and we have a couple different patterns going on. Okay, one of them is that, okay, these are the number of insects coming in 2021 and 2020. So yes, there's a difference between years. I'll get to that. But in both years, you see, again, they're coming in here and they're coming in hot in October, okay? And now you're talking on the order of 12,000 per tree. So much big difference in numbers coming in October. Several miles away at this Meadowwood site, you see something completely different. Yeah, they came in October in numbers in 2020, but in 2021, that shift was that peak was shifted. They came in earlier, okay? And so this is a cool site because here Brian's inserted these, put these poles in the ground, and he's treated them with deltamethrin netting, so anybody who climbs up there gets killed. And so, and he did the counts. And so if you look at that at this same site, they're not coming in to feed on this tree. This is like ambiently when they're moving. You see there's a difference slightly between males and females, but in 2018 and in 2019, that peak occurred, September, early October. If you look at 2021, it occurred a little bit later. And then, basically as you go down, these are subsequent years. So this is, 2020, you had an earlier peak and then a later peak. And then 2021, again, shifted a little bit later, but you see the numbers were declining. And so now we're seeing, okay, we have, is it the case that, if I want to go back here for a second, you heard Richard Blair yesterday, working at a vineyard saying that, okay, are these populations crashing? Why do we think so? Did they deplete their hosts? So are we seeing multi-year patterns? It's cool that we're able to record them. It's unusual you can get data from the same site for multiple years, but now what explains that? And so here, some of the stuff that I'm doing is really working on trying to measure aspects of lanternfly fitness from wild populations to really have like some kind of a series of benchmark indicators to really try to understand and tease out these patterns of variation. So we have lanternfly that we're measuring and I'm dissecting, collected across different hosts. Do they have different fitness levels? Again, these are like your cage-free lanternfly. They can move on to anything. And so do we see differences there? I also think it's really important for us to have a sense of what's going on in these wild populations to really kind of better interpret what's going on in field studies and semi-field studies. So here, what I'm showing you is that like with Laurie Nixon's work, where they've reared lanternfly on different plants and whatnot, that if you look at the area of yellow exposed for lanternfly in lab studies, that they're much less developed than in field studies. Does that mean, or in wild populations? And so does that mean we don't do the lab studies and field studies? No, but in order to really interpret them, we need to have a sense of what the true capacity is of wild populations. And so here, if like we're going to continue to do these, survivorship studies, and again, they might be artificial, but we have to add this other dimension that also looks at fitness to better interpret these things. So basically what we're trying to do is, all right, can we look at some change? If they're really feeding well on cucumber or other plants, then they should increase their weight, or maybe we can, take indices of their fat bodies or subsamples of these nutrient status indicators. In order to do that, you need to mark these guys, okay? And so we call this picture girls with hats. Okay, my lab shellacked these little numbers onto all these lanternflies so we could do this. And by the time we collected them out, we weighed them, and we started to put them on the plants, the shellac melted, or they clotted off each other, or what. So we still have to figure out how to do that marking. But kind of the point is that we need to add additional approaches, because there's some cool patterns there. We need to tease them out. So another thing we're doing in our lab is Holly, my postdoc, is doing EPG studies, where basically you run an electric circuit through the insect. It's tethered up here to a gold wire, and there's then a probe in the plant. You run this electrical current, lanternfly will fight you tooth and nail, they will fight Holly tooth and nail. But basically when they insert their mouth parts though into the plant, then you can measure waves that reflect different types of feeding behavior. We need to do the histology to kind of decode what these different spikes are, but this has been done with other insects. And you can see, you kill them, with the mouth parts in the plant, you do the histology and the microscopy. This is what Holly is an expert in. I'm very excited. And you see where they were feeding. And so you can see, okay, how much time are they spending salivating? How much time are they spending probing, inserting and drinking on phloem cells versus xylem cells, that kind of thing. And so again, we just need additional tools to really kind of couple with these other, with these studies to really understand feeding behavior. There's other things that folks are doing. Here's a different thing. They're feeding on cucumbers. So we want to look at these different hosts, or even different varieties, perhaps of grapes, to see, how exactly they're feeding. Are there some varieties, or are there some crops that make it a little bit harder for them to feed, or take a little bit longer to feed? So that's some of the stuff we're doing. Again, we're working all very collaboratively with other folks. Tracy's postdoc's doing some cool stuff with using, and I don't know if she'll talk about it, but using molecular gut content analyses to see what cage-free lanternfly are feeding on. Her postdoc Jim Helper's working on that. So a lot of really cool multiple approaches we can take to try to tease out these unusual patterns. Also, I just want to mention other things we're doing in my lab. We're looking at temperature variation across life stages. Hooping is going to show you some super cool stuff, with some thermal things. We want to know just, what are the thermal limits of lanternfly? We're about to put out or submit for publication a study characterizing what is the fungus, or what are the fungi that characterize sooty mold. And then for folks in the Helmus lab, we're finishing up some preliminary data on microsats that we can hand off to them that hopefully might help them figure out their population tracking of lanternfly. And so with that, I kind of gave you the bird's eye view of what I'm up to these days, and with all the folks in my lab who are wonderful. So thank you, all my lab and my extended lab members, Brian, John, Rost, and Dennis, and check out our SCRI project that's funded a lot of this work, but so has the Department of Agriculture for the state of Pennsylvania and Penn State. And so with that, I think I'm finished. Thank you.
All right, thanks, Julie. Yeah, I just saw there was one question I had that came up in the Q&A about the cucumber study. So it was asking, for the cucumber with the high SLF, indicating low yield, with the low yield based on yield, like grams per hectare, or marketable yield, like the size type.
Basically it was size, but it was also this was really just those 4 plants, so it was not scaled, you know.
Right.
And so we're going to do that. I need to look and see it. I can look that up with what she did, because I don't know if it had to do with also the number of cucumbers produced per plant, but I'll look that up and I'll write that in the Q&A.
Okay. And then just one more, sorry. Could cooling decreasing temperatures rather than the accumulation of warmer temperatures be the cue to move to alternate hosts and oviposit?
It could be day lengths. It's the other thing that really Dennis has been looking at. But yes, what's interesting is that one thing that I was wondering in terms of like that egg laying, is it the case that they essentially egg dump, that it gets, you know, close to it, you get a cold drop, you know, and then suddenly those eggs appear. But some of the data that Brian Walsh is collecting, where they're marking the egg masses, allows us to get a sense of when they're laid and how that works. So I don't have the answer to that yet, but Brian has the data to answer that. And so that's one of the things we're working on.
Okay, thanks, Julie.
Yep.
All right, so now we'll move on to Charles Bartlett as the next presenter.
All right, so do I have audio and can you see my slides?
Yes, the both.
Okay, very good. So I'm Charles Bartlett, University of Delaware, and I'm part of the project that is looking at development of biological control methods for spotted lanternfly, which is one of the objectives of the SCRI project. And I'm going to be presenting things at a very high level, kind of ignoring a lot of details and digressions that we could possibly get into. So just an overall look at things. So the development of biological control methods got started pretty early in this. After spotted lanternfly was detected, our main objectives are to support the discovery of biological control agents of the spotted lanternfly under the hypothesis that natural enemies reduce spotted lanternfly populations below harmful levels. And as I say, I'm just going to present kind of a high-level overview of where the circumstances lie. I'm going to put my credit slide right at the beginning. I need to point out that a lot of what I'm presenting on is not work that I've done myself or necessarily even work that I've directly overseen. My graduate student, Tyler Haggerty, is responsible for most of the actual work. And I have another grad student, or about to be grad student, Kat Williams. But a lot of what I'm presenting is actually work done by Hannah Broadley at USDA and Kim Holmer. So when we get down to questions, there might be some detailed questions that I won't be able to answer because it reflects their work, not mine. Okay. So in quest for biological control, USDA and partners started to foreign exploration right away. Spotted lanternfly, of course, was detected in 2014, and already in 2015, they were working at finding a biological control agent, since it struck some of us as fairly obvious that that would be what would be needed to get the spotted lanternfly under control. So foreign exploration was done between 2015 and 2018. Of course, the pandemic kind of stopped that process, but it was still being undertaken by our cooperators in China. In the process of that foreign exploration, 2 possible biological control agents were found. I'll look at each of those very briefly in just a moment. Both of these are currently in quarantine at USDA facilities. So the first of those 2 possible biocontrol agents is Anastatis orientalis. It is a chalcidoid wasp in the family Eupelmidae. It is a parasite of eggs and it was described as a egg parasite of the spotted lanternfly. To give you an idea of its efficacy in China, about 70% of egg masses that were surveyed were parasitized, and within those egg masses, about up to 33% egg parasitism. So it isn't necessarily something that's going to really take spotted lanternfly out, but it certainly is something that's going to slow it down a fair bit. This organism is being reared successfully at USDA facilities. And I wanted to point out this egg mass here. You can tell when there was emergence of a parasite rather than spotted lanternfly. The spotted lanternfly emergence shows these kind of elongate oval holes, where the wasp emergence, this is anastatus, is these distinctly circular holes. So you can look at a spent egg mass and determine whether there were parasites coming out of it, which is significant for some of our current work. So the second possible biocontrol agent is Dryaenus sinicus. It's in the family Dryaenidae. If you're not familiar with these creatures, they're very interesting. The females have these chelate front legs, because what they do is they hold on to a immature archanorhynch, in this case plant hopper, and lay an egg on it. So they need to hold on to it. And the males don't have that, but they're a larval parasitoid with the immature Dryanids being internal at first and subsequently developing into this external structure called a thylacium. So you can find plant hoppers and leaf hoppers immatures with these thylacium from our native Dryanids. Right now, the status of Dryaenus is that we're still working on improving rearing methods. And for that reason, I'm not going to talk that much further about this, other than to point out that when we get better rearing methods, then we're going to have to do host suitability studies with it. And that plays into what I've been doing. So as you probably know, prior to release, biocontrol agents must be studied to assess impacts on non-target species. And that's kind of the work that I've been doing primarily here at University of Delaware, where we've been collecting and rearing non-target, focal non-target species. This is mainly done by my grad student, Tyler Haggerty. And we rear these things and supply eggs to USDA as available and as needed. I have to point out that most of the species we're targeting are really very poorly known. So we have to figure out the techniques as we go along. I'm just going to give you kind of the idea of how, this gives you an idea of what our focal taxa have been. This slide is a little dated. We're targeting a large-bodied univoltine species that overwinter as eggs that are phylogenetically close to spotted lanternfly, and they lay eggs on above ground portion of plants. And that kind of description will exclude quite a few taxa. What I'm showing here are 8 families and 14 species that we reared here at the University of Delaware. As of a couple of years ago, we started coordinating a little more carefully with Lisa Tewksbury at University of Rhode Island, and her lab is doing the Heteroptera, and we expanded in the Lepidoptera as well. And my lab is principally doing the Archanorhynchus hosts. The check marks there indicate we've reared them successfully and obtained eggs that could subsequently be used for host suitability studies. We moved into some other groups of insects in part because some of the anastatus species parasitize those groups, and we needed to look at them and see if anastatus orientalis would. So to give you an idea of how this rearing process works, I'm going to show briefly 2 exemplar species. The first is Rhynchomitra microrhina. It's a dictyopharid, a fairly large and reasonably common dictyopharid in our area. We field collect species and right here on the university farm. Mostly we are successful in getting adults. We're not exactly sure what the immatures are doing, but the adults are not so hard to find. We move the captured specimens into a mesh cage in a greenhouse in a large storage bin planted with goldenrod and kept moist, and they survive just fine in those circumstances. We have found in the field they are found primarily on a plant called panicled aster, and we probably should try and rear them on panicled aster and see if that improves things any. We find they lay eggs on plants kind of loosely at nodes where they probably subsequently fall off into the soil. And we've been successful in overwintering them. And as long as the eggs are kept moist, we can have them overwinter. We've had our biggest problems with rearing species has been getting them to successfully overwinter. And when they emerge, particularly with the next species, they sometimes emerge too early, and we have to scramble to give them something to eat to keep them surviving. The other species I just wanted to highlight, just to give you an idea how this works. I'm certainly not going to go through the other species, is Publicia fuliginosa. That is a fulgura, just like the spotted lanternfly, and it has some life history characteristics that's pretty similar to the spotted lanternfly. This species is a more southern species, and so to get it in the field, we have to travel down to North Carolina. We've tried to go early in the season and collect early instar and nymphs, but the one year we did that, we got rained out by the ends of some hurricane. So we've stuck with going down just before the school year begins in August or early September. We found the Bladen County, North Carolina, where we can find them on winged sumac. And we only knew about the host just because of people that were familiar enough with these species to be able to take a stab at what they were found on. Interestingly, they're not on any just winged sumac. They're mostly on plants that are surrounded by weeds, and they're down in the base, hidden in the weeds. So they're not necessarily easy to find even then. The eggs of Publicia fuliginosa are laid in a very similar manner to spotted lanternfly. So in these groups covered with some sort of waxy material. So this has been one of our principal targets for host suitability studies. We've been supplying eggs of this species and others that we've reared for host suitability studies to USDA as needed. We have been successful in overwintering the species, but one little problem we've had is that the immatures are emerging too early. In fact, we have immatures of Publicia fuliginosa emerging right now, which is a little bit of a problem since we have to find plants for them to eat. Okay, so back to our potential biocontrol agent. The rearing of Anastatis orientalis discovered some fairly puzzling life history characteristics and some variability in the life history characteristics, which led to the suspicion that we're dealing with not a uniform entity, but multiple entities. And so Hannah Broadley's lab sought and found 4 haplotypes that they recognize, haplotypes A through D. And these haplotypes vary in their biology and some other details. And so we now have to look at this not as one species, but as kind of theoretical 4 species or 4 populations with different biological characters. We're working on establishing isolines for the 4 haplotypes. There is 3 of them that are reasonably good shape. I should say the molecular work was performed by Yunki Wu of USDA. So what our results of the non-target host suitability studies have been maybe a little discouraging at some level. These are the results for no choice testing of haplotype C. And there's several parts of this, whether they attack the species at all, and whether they successfully emerge if they do attack the species, and whether the emerging wasps are males or females. If they're just males, that means maybe they're not doing so well on that alternate host, but if they're females, then possibly they can survive on the alternate host, which is not what we want to hear. So for haplotype C, the results thus far suggest that it is not sufficiently host specific. And we're currently looking at haplotypes D and B, which may ultimately prove to be more suitable. But I don't have those results right now, and I don't think they're sufficiently well advanced. But this circumstance has led us to a little more interest in looking at what's going on in the field. And there's been some tantalizing reports of spotted lanternfly egg masses being attacked by species out in the field. So perhaps the spotted lanternfly is beginning to recruit parasites. And you see here 3 egg masses, 2 of which have a wasp sitting on them, which looks kind of suspiciously like Anastatus. And over on the right is a egg mass that has some spotted lanternflies emerge, but also a wasp as well. So we're seeing some evidence of parasitism of spotted lanternfly in the field. And also in 2017, Hoover reported that the inserted Insertus cavani, which was brought in for gypsy moth control, was found parasitizing spotted lanternfly eggs. So last year, Hannah Broadley's lab collected a lot of egg masses, 720 egg masses, to monitor for recruitment of biocontrol agents, native wasps, and they, out of 720 egg masses, they got 4 adult wasps that are tentatively identified as an inserted, a coleonid, and a species of dimorphic Megaspilidae. But this parasitism rate in the field from recruited parasites is extremely low, only about 0.7% of egg masses and about 0.04% of individual eggs. There was also 2 more wasps detected using dissections. So beyond these 4, there's some evidence of some some Anastatus activity on these egg masses. Some of the wasps that were photographed associated with egg masses look suspiciously like Anastatus. And so there was some work done to extract DNA from parasitized eggs where the wasp has emerged or the egg was found to be parasitized, to find out who it might be. And a species of Anastatus was found in this. Not entirely sure of what it is, but based in comparison the sequences that we had extracted from Anastatus reduvii, in kind of an effort to begin understanding our native Anastatus a little better, there's a 96.6% match with Anastatus reduvii, suggesting at least it is that species or a close relative. So it appears that the egg masses are beginning to recruit some native parasitoids, but perhaps not at a high enough level to have much effect on the populations. So that, I'm going to just briefly comment on what we're beginning to do now. This spring, or this late winter, I guess, we're going out and collecting egg masses right here at the University of Delaware. So far, we've collected about 250 egg masses. In fact, my grad students are out right now doing this. They're scraping them, not chipping them, and mainly that's to make it a little easier to get permission to do this sort of work. These eggs are being stored in a greenhouse situation, and we're going to be monitoring them for the possible emergence of parasitoids. Hope hoping we find some additional targets that we can look at. So that's where we're at. We have 2 possible control agents from China, Anastatus orientalis, with 2 haplotypes. We're still under investigation. Haplotype C is not suitable. We're still learning to rear Dryaenus cyanicus. And there's some tantalizing hints that spotted lanternfly is recruiting parasites in the wild, but still not enough to do much damage to the population. And we're still looking into that further. All right, that's what I have for you today. Thanks.
All right, thanks, Charles. Yeah, I just saw the I just see one question in there at this time. There's a person asking, what's the difference between scraped and chipped egg masses, and how does it affect rearing?
So ideally, we would take the whole egg mass intact. And to do that, you have to take what the egg mass is on. So if it is laying its eggs on the bark of a red maple, then you would have to chip that whole piece of bark away. And of course, that damages the tree, and you're not going to, parks and homeowners are not really going to be so eager for you to do that. So if you just scrape the egg mass off and track it that way, it destroys kind of the integrity of the egg mass, but we're operating on the assumption that we would still get emergence of parasitoids from an egg mass like that's been scraped off as opposed to chipped off.
Okay, thanks, Charles.
All right.
All right, so our next presenter is with USDA Agricultural Research Service. So it's Dr. Tracy Lesky. I think you got your presentation up and ready to go, Tracy. Or it was up, sorry.
Oh, it was up, okay. Let me do it again, sorry.
All right.
Don't know what I did. Can you see it now, Greg?
It's loading, yeah, it's up now. Yeah.
Okay, all right, Greg, thank you and everyone of the organizers for the invitation to speak today. And my goal is just to provide you with some updates on some of our projects under the SCRI grant, and also just acknowledge that we have many collaborators outside my group, that including Julie Urban, Ann Nielsen, Heather Leach, that have contributed to the studies that I'm going to talk about. So the first set of studies that I'll just highlight are the work that we did under the area of host plants and specialty crops. And these are just some of the sub-objectives from the grant that we are trying to address through the studies that I'm going to present. But the thing to keep in mind about spotted lanternfly and specialty crops is that it is an invasive landscape level pest, because not only can lanternfly, as we know, be feeding and reproducing in native or invasive woody hosts outside specialty crop production areas, but they also invade and move into vulnerable specialty crops such as vineyards, as we've heard a lot about over the last 2 days. So a couple of things I want to highlight are some data from some published and soon to be submitted studies on really trying to understand 2 questions. In this case, what single specialty crop and wild host plant diets can support SLF over a 2-week time period for nymphs as well as adults? And then a longer study where we looked at how does single and mixed host diets of specialty crop and wild host plants affect overall SLF development and survivorship. And so from this study led by Laura Nixon, where we had a number of different hosts, including apple, black cherry, black locust, black walnut, hackberry, mulberry, peach, sugar maple, tree of heaven, and white oak, if you look at survivorship over time of early instars, late instars, and adults, we do see significant differences. And as we've seen with other studies, the early instars can feed on a lot of things and they have reasonable survivorship. But as they develop, a lot of these single hosts, with the exception of tree of heaven and black walnut, cannot support survivorship over 2 weeks. And by the time we reach adults, only tree of heaven could. Then for that second question, looking at development on single or mixed diets, this is the percent of the cohorts that survive from first instar to adult. So you can see that Tree of Heaven or Tree of Heaven plus mixed diets yielded surviving adults, whereas apple, grape, and in this case, this was rotundifolia, a muscadine grape, and peach did not. And black walnut alone did support development and survivorship to the adult stage. However, if you look at their survivorship in the adult stage, you can see that the black walnut group, when they were only provided black walnut, did not survive. So this led to a series of studies that were just about ready to prepare for submission, where we wanted to kind of take this next step and ask similar questions, first with woody vine hosts, and asking the same kind of thing: can they survive over a 2-week period on, again, this is a rotundifolia or hops or kiwi, and compare this with Tree of Heaven. And so this was over 2 years, and we looked again at early, mid, and late instars, early and late instars, as well as adults. And so with the early instars, you can see overall most of the hosts supported survivorship pretty well, but not the kiwi, probably because the vines are very hairy and they couldn't access the food. Whereas late instars, all of them did okay. But by the time we reach the adult stage, you can see that declines in survivorships for grapes, hops, and kiwi compared with Tree of Heaven. And this then led to another study asking the same kind of question about development and survivorship. How well could they survive on grape-based or black walnut-based mixed diets? In this case, we use Vitis vinifera as the anchor host, along with apple, peach, black walnut, or silver maple, or Tree of Heaven as the positive control, and then the same with black walnut. And what was interesting about this is the big differences we saw when you had Vitis vinifera as one of the hosts compared with black walnut, where we saw greater survivorship and development when Vitis vinifera was included as one of the hosts versus black walnut. And if you look at the overall numbers of the individuals that reached the adult stage, you can see that these grape-based diets were, as a primary host, were far superior to those with black walnut, unless you had Tree of Heaven in the mix. But definitely Vitis vinifera appears to support development much better than Vitis rotundifolia, the muscadine grape. And so this is leading to a study that Joanna Elsenson is going to be conducting this next year in our quarantine greenhouse, where we're going to look at how different Vitis species affect survivorship and development. And this is based on the fact that with Vitis vinifera, 26% of all individuals on that single host reached adulthood versus 6% only reaching 4th instar for the muscadine grape. So these are the different species of Vitis that we will be evaluating, and we will include Tree of Heaven as a positive control. The other thing that we're doing relative to host is using gut content analysis to really understand more about their host use patterns. This is based on some work by our colleague Rodney Cooper, who looked at spotted lanternfly and using the techniques in his lab to evaluate gut contents and the plant DNA. And so from there, my postdoc, Jim Hepler, is going to be asking a couple of key questions using this gut content technique. How does the host range and use by SLF change between life stages and throughout the season? And are there any detectable differences in gut contents from those mass dispersing adults? So he's already started collecting, and he's got the lab up and running with the gut content work. And we'll be processing samples from last year, as well as doing additional collections this year. But ultimately, we hope that we can understand which host plants are important and when throughout the season, so we can improve scouting and management efforts. And also with this mass dispersing, can it help predict the risk of SLF invasion or just the possibility of a mass dispersal event? So we'll leave host plants now and move on to mating and dispersal behavior. And so we have one study that I'll talk about that's really at this point just about ready to be submitted as well. And the question was, what behaviors are expressed and how do they change over the course of the season when we have these adults that interact in groups. This is led by my postdoc, Joanna Elsenson, and also how does group size affect the behaviors observed? So this work was done in 2020 and 2021. In 2020, we just did these observations through September and October. And then last year, we were able to do them from August through November. And the general behaviors included feeding, mating, walking, and resting, but they were broken down further into courtship, mating competition, mating, feeding, and feeding competition, walking up, walking down, and resting. And so this is just a summary of the results, and there will be more to this when we do submit this, but you can see here are the different behaviors: feeding, walking up, walking down, courtship, feeding and mating competition, resting, and then mating. And so feeding is definitely, not surprisingly, a major activity, but we see this neat little kind of dip in October. And this is when mating is observed. That's that red at the top. Feeding competition was most prevalent early, but it occurred more frequently in larger groups. And we see that courtship began in September as well. So I think that this will be helpful data as we try to understand more about their behavior and how they interact with each other, and possibly lead to some new opportunities for olfactory attractants as well. The other study that I wanted to talk about relative to Joanna's work, and she talked a little bit about this yesterday, asking what factors influence SLF retention and dislodgement on vehicles. And Joanna recognized when she was going through the literature that there really were no empirical studies on insect adhesion to various modes of transportation. And so working with our engineering group, we have this laminar flow fan that Joanna and folks have been evaluating. And so from this work, we're really starting to see that SLF is actually, they can hang on quite well. And in fact, all materials that Joanna evaluated had some SLF that did not dislodge at the maximum wind speeds, which is about 55 miles an hour. And she's starting to see some differences among locations on vehicles and the different kinds of materials and weather conditions. So this will continue in 2022. And then finally, our last sort of movement study, this is going to be work that Jim Hepler is going to be doing, is trying to understand if we can better relate flight capacity and behavior to the physiological state of adults. And so based on the kinds of work that we've done over the years with brown marmorated stink bug, with flight mills, with harmonic radar, direct observations, we're going to begin this work with these techniques in 2022. Moving on to monitoring and tools and chemical ecology. This is another area that we spent a lot of time working on. And so I'm just going to highlight a couple of the studies that we have currently. One is on optimizing trap deployment. There has been a lot of work by our colleagues at APHIS, by our group, and as well as others, to try to develop traps that work well in terms of captures, as well as just trying to reduce non-targets. And so this is a study we conducted in 2021 and 2022 to try to learn what are the best ways to deploy these traps. And so we had a couple of different factors we looked at: the height of the traps, whether deploying them at 0.5 meter or 1 meter above the ground would make a difference in overall captures. And this was simply based on some of the original dispersal work that we conducted, showing that sometimes, especially for the adults, they don't always want to walk the entire way up the tree and be captured in traps. They'll sit below and feed. We also looked at trap sample interval with traps checked at 1, 2, and 3 week intervals. Host tree deployment. If you deploy these on tree of heaven or other common deciduous hosts, what do captures look like, to try to offer some flexibility? And then just relative densities of SLF. And in this case, we looked at sticky bands, this USDA jar top, circle trap, and bag traps deployed at high and low relative densities. And so this was led by Laura Nixon. And so if you look at the trap height data, this is 2020 and 2021 combined, you can see that there were no significant differences, but 1 meter height appears to be a little bit better numerically. And so that's probably what we would recommend going with. For the sample interval, you can see again, we had a lot of variation, no significant differences, I believe, but the issue is, and we'll talk about it a little more, is that we do see a lot of sample degradation over time. With host trees over the 2 years of our study, we did not surprisingly see that greatest captures for both nymphs and adults were on Tree of Heaven, followed by black walnut. But you can see that all of these trees yielded some level of captures. And so, you know, if a Tree of Heaven isn't available, it means that, you know, there is opportunity to still trap on some of these other SLF hosts. When we look at population densities for sticky bands and modified circle traps in 2020, and the replaceable bag, the sticky band, or the modified circle trap in 2021, a couple of things. One is all of these traps did capture individuals, and under low population densities, we did see better overall performance from the circle traps, particularly for adults. But one thing that was really interesting to me looking at these data is the fact that under low population densities, we were capturing more adults, but when you get under high population densities, we're getting more nymphs. And so just something to think about, you know, in terms of trapping and what to expect in certain sites. So in terms of the key results and implications, again, circle trap height, we didn't see significant differences, but probably 1 meter is still what we recommend. Trap sample interval didn't necessarily affect the overall captures, but we did see degradation and what we call the yuck factor and likelihood of non-target occlusion, particularly when you have periodical cicadas filling your traps in the early season. But we would recommend weekly sampling if possible. Host tree deployment, again, Tree of Heaven followed by black walnut, but all of them yielded captures, so that does offer some flexibility. And finally, in terms of relative density, all of the trap designs worked well, but the bag trap did lead to degraded specimens, and sticky traps, obviously, we get a lot of non-target captures. The last study I'll highlight, this is really just getting going, is asking the question, can we identify novel attractants for SLS based on behavioral observations made under field conditions of what we call hot trees? And so where we see trees that consistently yield lots of spotted lanternfly feeding on them. And so our question was, could we make our own hot trees? And so working with our breeding program, they have identified this species or this strain of cladosporium fungus that induces plant growth promotion, early flowering, and increased fruit yield in other plants. And it also increases sugar uptake. And so they were able to put Tree of Heaven into tissue culture and expose these plants to this fungus. That's how it's exposed. And they did see increased growth from these plants, and that they were induced. And so we were able to take them to the field just to see if we could see any differences. But you can see when you induce these plants, they grow more quickly, they produce more fruit, and they produce more roots. And so I thought, well, maybe we can make a hot tree for SLF. And if we can, then we can start collecting volatiles and compare them with uninduced trees, and as well as our GCEAD work. And so last year, this was very preliminary, but we went out, and this is a Tree of Heaven that was induced versus the control. And what we did was just put out sentinel trees and do weekly counts. So we didn't know what was going on in between, but you can see that early in the season, when we first put these trees out, we saw significantly greater numbers feeding on these trees. Now, over time, they started to decline, and I suspect this is because of increased feeding. And so we replaced them halfway through, and we saw an increased peak again. But again, there was just a lot of variation, and we weren't necessarily, this was, like I said, kind of preliminary, but we're kind of excited about it. I think there's something to this, and I look forward to exploring this further in 2022. Laura Nixon is leading this work. And so with that, I will conclude and just say thank you to everyone in my lab group, as well as my colleagues. And if there are any questions, I can take them now or later, Greg, whatever works best.
Okay, thanks, Tracy. Yeah, there's There's a question on with 1 meter off the ground trap deployment, did you have any animal interference? And were the locations in publicly accessible zones? And did you have any human interference?
So most of our traps are deployed in not necessarily places humans would go, except for one location, which is a park-like location. We never really had any trouble, but that's because I think, especially with the Extension Service in Virginia and West Virginia, have done an excellent job of really educating the public about why these are present, and you know, and so we haven't had any issues that way. In terms of animals, so far nothing too serious. Occasionally, you know, especially with the circle traps, we don't really have too many issues.
Okay, thanks. Yeah, and I actually clicked on another question that was on here, but other people can answer too, but I can throw out what I know on it about if there was any research on whether native species like assassin bugs are preying on SLF, but I don't know of any like specific research on it. I've just seen a lot of anecdotal evidence and photos, mostly of adults of assassin bugs and wheel bugs and praying mantises also feeding on them. But you know, again, they're kind of categorized as generalists, so they're not expected to have like a huge impact or really reduced populations, but they haven't found feeding on them. You know, and they'll get trapped in spider webs too, and yellow jackets will take advantage of them also. But yeah, that's all I have, unless somebody else has other information, but I'm not aware of any specific research on other native species that are that are predators on SLF. Okay, so we'll move to our next presenter, which is Kelly Hoover with Penn State.
Thanks, Greg. And thank you all for tuning in. What I'm going to be talking about today, for the most part, is related to the SCRI grant objectives. And I'd like to acknowledge my collaborators there with the asterisk, who are plant physiologists and were a big help on these some of these studies. So what I'm going to address today are 3 research questions. Is tree of heaven a required host? Does spotted lanternfly affect ecophysiology, carbon assimilation, and or growth of common hardwood trees that are used in forests? And how far do nymphs disperse in contiguous forest? So this is sort of leading up to the section coming up on forestry. So in this study, we set up 10 plots at Emily Swackhammer's farm in Berks County, that each of our plots had silver maple and willow. We also planted black walnuts, but they didn't do very well, so they really weren't fed on. And half of these plots had tree of heaven and 5 had river birch instead. And in the first year, we started with egg masses that hatched into neonates. Second year, we started with third instars reared in pop-ups that still did not have access to tree of heaven, or did. And we monitored the time from egg to adult and each life stage. And it took about a week longer for the insects to reach adulthood without tree of heaven, and about 2 weeks longer to begin laying eggs. And the egg mass ratios with Ailanthus were about 7-fold more in both years. The other thing was we hatched offspring from the first year parents and reared them all on tree of heaven. And we saw equal hatch and equal development time. So there were no carryover effects into the next generation. This has all been published, so I'm going through it kind of quickly. Then we also looked at host preference, or if you prefer, host association, although we were seeing a lot of feeding on these trees. In the presence of Ailanthus, the nymphs did indeed prefer tree of heaven, and without Ailanthus, they equally fed on willow and silver maple. The adults preferred tree of heaven, but just as much as many of them fed on silver maple. That may have been due to the fact that Tree of Heaven may have been getting an awful lot of pressure the whole season, and so may have been reducing in quality for the lanternfly adults. Without Ailanthus, they strongly preferred silver maple. I'm sorry, I have a cat that makes a lot of noise when she wants to eat. Then we did several studies at a common garden, also in Berks County, where we planted about 800 trees on 2 acres, and we had 4 tree species, and I'll be talking about different ones as we go along. Red maple and silver maple were tested in the first year. We tried nymphs at 3 different densities and saw no impact on tree parameters. But when we put adults on at just 1 density, we just wanted to see if there would be any effect. And we measured gas exchange and carbohydrates. I also want to tell you that in every experiment we've ever done, survivorship is greater on silver than red maple for all life stages. So it might explain why we see them more often on silver. It's just not as common in the field. So in the sleeve cages, we got pretty rapid suppression of photosynthesis. There were no impacts on carbohydrates, but this was only a 10-day study. We also saw rapid reductions in transpiration, and the stomates closed pretty quickly. So then we moved to whole tree enclosures on our larger trees in the common garden. And we wanted to do this because in the field, the adults are actually feeding on the trunks. The problem the year before is we didn't have big enough trees to really confine adults on whole trees. So adult feeding on these trees did reduce photosynthesis over time. Time is on the X and photosynthetic rate is on the Y. And the reduction was especially strong, believe it or not, at the moderate density, even more so than at the highest density. In fact, at the highest density, it wasn't significantly different. We saw a similar effect 2 weeks after feeding on Tree of Heaven, and transpiration also declined markedly on Tree of Heaven. In particular, again, at the moderate density. Silver maple photosynthesis, stomatal conductance, and transpiration were all reduced, especially at the highest density, but not at the lowest density. So back to Makanji. While we had these multi-tree enclosures set up, we decided to go ahead and measure gas exchange and also look at carbohydrates. So in these multi-tree enclosures, we did a bunch of different metrics: gas exchange, root and stem sampling for carbohydrates, nutrient concentrations, and we also measured trunk diameters. And we did have some controls that were in these enclosures that are off to the right that you can't see, that were also measured. For all species except river birch, which hardly got touched, they rarely fed on it, photosynthesis did decline with increasing lanternfly pressure. These insects were on these trees from May 22nd to August 1st at this point. So this was done on August 1st. And you can see that river birch is the only one that didn't have an effect. By October in the following year, I'm just showing you some dates. We did like 3 or 4 dates every year. Photosynthesis did decline as a function of density, especially for silver maple, which is in the red right here. And what was different between 2019 that I showed you at first, which was the first year of the study, and 2020 was in this year, we had started with third instars. So they hadn't been fed on as long as they were in the year previous. So in summary, because I don't have time to show you everything, the impacts of lanternfly in these multi-tree enclosures were that carbon assimilation and stomatal conductance did decline for all species except river birch with increasing density, especially with life stage. The available soluble glucose in stems was enhanced with increasing densities, believe it or not, for willow and silver maple, but it was unaffected in tree of heaven or river birch, of course. The starch in stems, however, was reduced with increasing adult densities, except for river birch, yes, and tree of heaven at the highest density. Roots are still being analyzed for carbohydrates and nitrogen for all of our studies. It's, Julie, you will find this out. It is very difficult to get access to a freeze dryer in our college because 2 of them are so old you can't get new parts and they're broken. And there's everybody else in the whole college wanting to use the 2 that are left. Nitrogen was reduced for tree of heaven in stems and leaves. Phosphorus and potassium were reduced for all tree species except river birch at increasing densities. Trunk diameter growth was reduced after 2 years of feeding pressure. So if you look at the diameter increase in 2019 versus 2020, and this is in reference to the controls, they grew more in the first year than they did in the second year after 2 years of feeding pressure. The only place we didn't see that was in river birch, which is interesting because in the field, river birch is attractive to adults, but it's the cultivars more like Duraheat and Heritage. This is not one of those. It has a different bark color and it doesn't seem to be very attractive to lanternfly. So something kind of fun that was happening that year that I wanted to show you, the second year, is that this was one of our enclosures, and those are lanternfly adults that are all over that cage. They were only on that cage and no other cages. So for some reason, this tree on the edge here, which was a willow, was a hot tree. Because what they're doing is they're feeding through the mesh on the branches that are oppressed against the screening or the mesh. Also, several people have shown that when lanternflies feed on grape or maple for a period of time, if you peel back the bark, you can see damaged cells. So there is definitely damage going on. And in fact, Christina Rosa talked yesterday about how looking at grape, they saw upregulation of many genes that are in response to injury. The last thing I'm going to discuss is host availability impacts dispersal, because I'm going to talk about dispersal and how the availability of hosts can affect that. We already know what the preferred hosts are, and in forests, well, willows aren't necessarily in forests, but these are the sorts of suitable hosts that you find in forests. And my guide about is something going to be a host plant or not, especially to early nymphs. If it's an angiosperm, it's always possible. We've even seen them feeding on poison ivy, excuse me. So we did a dispersal study. This has been published in a contiguous forest, Musser Scout Reservation. We did a release of each stage of nymphs marked with different colors of fluorescent powder. And we released about 2,000 of them in the center of 12 spikes going out from the center. They were 50-meter transects. We did our searches at night with UV flashlights and handheld lasers. And when we would find a lanternfly, we would drop down a marker, and then the next day in the daylight, measure the distance from the detected nymph to the transect line. And this is some of what you see. With the red marker during the day, they look like this, and at night, they look like this. The first few days, most nymphs were found about 5 meters away up in a black gum tree that was slightly uphill. They had a tendency to walk uphill, which was interesting. But on average, their dispersal was about 17 meters after 7 days. This is what we saw in our transcripts at day 3 of third and fourth instars. So the greens are the fourths and the reds are the thirds. So we were seeing both of them. And we did find a third instar 65 meters out from the center. This is also consistent with what we saw when we tried to study the impact of aerial spraying of different insecticides at Blue Marsh. We had a ton of movement of nymphs from plot to plot, which made it very hard to see efficacy of the treatments. But the median dispersal distance was the furthest for third instars. So in summary, Tree of Heaven is not required for development or reproduction, but fitness is reduced, and there is a delay in development rate and certainly an impact on fecundity, but fertility was not affected. And as Tracy has talked about and a few other folks, diet mixing was very evidently favorable for being able to reach adulthood without ever seeing Tree of Heaven. Gas exchange and carbon assimilation can be reduced in hardwood hosts with prolonged feeding. In the field, we don't usually see very prolonged feeding on these hosts that are not Ailanthus, except for grape. And usually the impacts we saw were in a density dependent manner. And with just feeding by adults, the effects were minimal if it wasn't adults. Nymphs seem to have minimal effect. In forests without many suitable hosts and not many understory plants like the forest area we used, nymphs will move as far as they need to find suitable hosts. We also had them molting within a week, so we were no longer able to find them most of the time after the first week. And initially, they don't move very far. They climb up the first thing they seem to come across. In the field, we rarely see tree death, and this is consistent with what our results are of any tree species except Tree of Heaven, and maybe young saplings, probably in that case from multiple stressors. And with that, I have a lot of people to thank, and especially the crew in the middle. And I can take any questions if we have time. Thank you.
All right, thanks, Kelly. Yeah,
Sure.
again, like the rest of the presenters, a lot of information in a short amount of time.
Yeah, well, wait till you see how long this paper on plant responses is going to be.
Well,
Most of the data is still being analyzed. I got some of these slides this morning because they were being worked, the data was still being worked on yesterday.
Okay.
A lot of data.
Well, there's a couple of questions in here that either like you could answer or one of the other presenters. So the first one is, I know this has come up before too. Are there different definitions for hot tree?
Well, Tracy can say what she wants to say, but what I would say is what we call a hot tree is when we see tons of lanternflies on a specific tree, when there's other of the same host that look exactly alike right next to it, and there's hardly anybody on it. So that's what we're calling a hot tree.
And that's exactly how we interpret a hot tree as well, and that we can go back for a period of time and continue to see the same hotness. So yeah.
And what I would add is that despite how it sounds, we're not insane, which is kind of how it sounded at first. And so this characterization that certain individual trees, when you're comparing trees of the same species, certain individual trees continue to attract high numbers of lanternfly was published in a paper in Environmental Entomology led by Charlie Mason, so Mason et al. And that's where it showed it, like those trees were treated with insecticide, and you can see they kept coming in. So we have data to demonstrate this actually happens.
Okay.
And we've all seen it in the field, yeah.
And there's two more questions in the Q&A. If any of you would like to answer those, that would be great. But I see we're at 11:25. I thought we had till 11:30, but double checking the agenda, it's 11:25 is when we're supposed to end and
Okay,
go to break.
Greg, I can take the first one because we're working on that.
Okay.
I
Take the
have a
second
graduate,
one.
yeah, I have a grad student working on sequestration of toxic chemicals from Tree of Heaven. This specifically, she's focused on the quasinoids, which are bitter tasting compounds. And we do see some effect on bird predation, but not on arthropod predation. So we still see hundreds and hundreds of reports. We did this through a Facebook citizen science project of arthropods feeding on spotted lanternfly, and in experiments we've done where we've taken insects reared without ever seeing Tree of Heaven versus those that did. It affected bird feeding, but it did not impact spiders or wheel bugs or praying mantises or other things like that. They still fed on them. And we're
Okay.
now starting to work on predation in the field by native natural enemies.
Okay. Thanks, Kelly.
Sure.
So yeah, so we'll go to break now. I think then we're supposed to be back at 11:35. But yeah, if you want to add any more information into the Q&A section and the answers there, that would be great.
Sure.
Like to any of the presenters. All right, and thanks for the four presenters this morning and the information you had. But I believe 11:35 is when we're supposed to come back according to the agenda. All right, thanks.
Hey, Kelly.
Yeah.
So here's a question for you, and we're going to see what we get from our gut content analysis. Julie, feel free to weigh in, but sycamore, and Ann talked about this briefly yesterday. Sycamore, we have a lot of sycamore, and we're seeing them. What's your take?
We've seen large populations on the sycamore when it was super wet. We had that really, really wet year. Was that 2018 or 2019 up here? And they were not on the maples. The roots of maples were all in water, but there were some sycamores on some higher ground, and they were all over the sycamores. Brian, it was with me when we saw that.
Okay. Yeah, because we're seeing it here. And so I was just curious what you guys were seeing. Like they're definitely using sycamore. We have a lot of sycamore. So
Well,
yeah.
This is what really struck me in like looking in more detail. Like I looked up like rereading like the Duchesne paper and stuff. I forget what the genus is, but basically the BB tree, right?
Yes.
That introduced, we don't have that here. It's never come up as a report. And it's like, oh my gosh, the relative abundances of these other alternative hosts, especially even things we don't think of like that, or at least I don't think of, that you're just going to continue to see these different kind of shifts and weird things, I think, as it expands. And so it could just be, you're also going to see higher proportions on sycamore if there's more where you are, which is what's so scary about it, I think.
Yeah, I just answered the question about homeowners who are concerned about lanternfly feeding on their trees and shrubs and flowers. And the bottom line is that they're not going to stay there long enough probably to do any harm unless it's Tree of Heaven. You'll see them on roses, and sometimes they'll stay there for several instars, but I haven't ever seen roses negatively affected. What seems to happen even like on black walnut is you can get huge populations on branches and it'll kill the branch. You see the branch wilt and the tree just drops it. It sacrifices that branch, but the whole tree itself is not impacted. It seems to be able to compensate, especially older trees. Younger trees have more trouble.
Kelly, your answer, I added to your answer then too, because like in the chat, Brian's like, it's all relative to what's available, which is totally what it is. And so I think the management guide like has that really well covered, and the stuff that Brian, he'll probably share with his treatment, but it's just the idea that especially with the nymphs, they might move in, but they're not going to come in high numbers and stay like the adults are. So don't pretend you can keep them off your property. But the management guide that you all did, Amy, Brian, Heather, and Emily, is really good with that.
And I'm going to talk about this afternoon.
Okay,
great,
cool.
fantastic.
I agree. I think the only trees I think it killed is we had some Atlantis tree actually been killed.
Yeah,
in the field. But other species, as just as Kelly, as you said, you got a huge numbers on the one of trees, and I don't think they stay long enough on other trees. So
Right.
Yeah. And like, I also wonder, that's what I wonder with these multi-year patterns. Like, did they just hammer, the ailanthus and some of those trees from 2013 to into 2017? And then that's what we knew they were flying, right, in other years, but then that big movement, perhaps of a greater bulk of a population was because they had just kind of hammered those. And it's almost like what we're seeing again now. It's like a saltatory kind of movement out, I don't know. Yes, the Tetridium, Doug. That's exactly what it is. Yeah,
The
thank you.
BB tree,
I
eh?
couldn't remember.
So Chad asked, is there any feeding on American sweet gum? Yes. Would offspring reared on host plants in the absence of tree of heaven be able to maintain fitness over several generations on those hosts? I don't know. We haven't tried that. We mostly just wanted to see if we found fitness effects from the prior generation, but it's a good question. Somebody should try testing that.
Well, and I think the bigger problem is like trying to maintain fitness across multiple generations, rearing them in captivity, period, regardless.
Period.
That's
That's
the
right.
point.
It's
That
tough.
was my yellow belly things. They're
They're always going to be smaller when you rear them in captivity. They just are not as robust as when they're wild caught. Branch mortality in the lower branches, we saw it upper branches, lower branches, middle branches. That was at Musser. They were on every black walnut that we saw in 2018, and most of the branches. Community gardens and urban farms. I'm going to let our extension folk address that. Yeah, grape is quite susceptible. That is absolutely true. I mean, it's more susceptible. There's no doubt that the impacts are much more straightforward than what we're seeing on hardwood trees.
And I think like whether or not there's varietal differences, particularly why it's so interesting what Tracy's group has done in looking at the muscadine, because my understanding there is that muscadine has like a natural resistance to Pierce's disease and that kind of thing. So that's why I'm interested for us to look at EPG to see how easy and how hard it is and what those variations are in different varieties to try to figure out what plant traits confer greater like resistance.
Right. Well, the same goes for hardwood trees. There's different cultivars, like even among the red maples, there's a lot of difference between the Freemani hybrids versus the straight red maple, swamp maple.
All right, well, I need to go to class. Bye, everybody.
Have
I'll watch
fun.
the recording.
Bye, Julie, have fun.
Bye.
Your last day, right?
Yep.
Yippee!
Yay. Bye. Great job, everybody. Thank you so much.